Hi, everyone. Thank you so much for having me. So today I'm going to be presenting on my work called AMAISE, which stands for a machine learning approach to index-free sequence enrichment. This work was done jointly with Piyush Ranjan, John Arp Downward, Robert Dixon, and my advisor, Jenna Weems, and it was published in Nature Communications Biology in June of 2022. Okay, so first I'm going to be getting into some background related work related to AMAISE. So the ultimate goal of our work is to improve infectious disease diagnosis by developing a pathogen identification tool that is accurate to ensure that correct treatment can be given, fast to ensure that the treatment can be given in a timely manner, and cheap and efficient to ensure the accessibility of the method and the ability of it to be used at the bedside. So traditionally, to diagnose infectious diseases, clinicians have used culture-based methods, which involves taking a sample from a patient, either stool, saliva, or blood, mixing the sample with culture media and waiting for growth, and then doing pathogen identification via biological tests. There are a couple drawbacks to using these culture-based methods. First, they can be inaccurate because not all pathogens can be detected using this method, and pathogen detection is necessary for treatment. And this is because not all pathogens can grow when mixed with the culture media. These methods can also be slow because it can take several days to detect growth, and this slowness can negatively affect a patient's ability to recover. So to alleviate some of these issues, people have started to use metagenomics for disease diagnosis. So metagenomics is the study of the metagenome, which is the collective DNA from a clinical sample. And the way that people will use metagenomics for disease diagnosis are that they would take the same sample from a patient, the stool, saliva, or blood, but instead of mixing it with culture media, they would input it into a DNA sequencer and get metagenomic sequencing output, which is all of the DNA in the sample. And then they would use a metagenomic classification method to do the pathogen classification. And metagenomic classification maps DNA to a classification label, and existing metagenomic classification methods do this using reference databases. So as you can see here, we have the metagenomic sequencing output that we input into this metagenomic classification method that uses a reference database to do the pathogen classification. So the problem with using metagenomics for disease diagnosis are that there can be copious host-derived sequences in a sample. And here I have a couple of examples of microbiome compositions from a human where in the pie chart, the red indicates the percentage of the sequences in the microbiome that are from host, and in blue we have the percentage of sequences that are from a microbe. So the gut, as you can see here, is evenly split. There are 50% host sequences and 50% microbial sequences in the microbiome, but the lung can be can contain copious host-derived sequences compared to the microbial sequences. And here's an example of what a lung microbiome could look like with the copious host-derived sequences. And so microbial sequences are informative for pathogen detection because they could contain the pathogen-derived sequences, but the host sequences are not informative for pathogen detection. And in this case, host is the patient from which the sample was collected. So copious host-derived sequences can cause downstream classification methods to be inaccurate, slow, and costly. Inaccurate because these methods are not especially sensitive to noisy host sequences, so they can be inaccurate at classifying those sequences. Slow because it causes these methods to classify a lot of uninformative sequences. The host sequences, as you remember, are not informative to doing the pathogen identification. And also it causes these methods to be costly because they have to store large amounts of host genomes to classify the host sequences. And typically host genomes are much larger than microbial genomes. So this can add a lot of storage and RAM requirements to these methods. So to alleviate the aforementioned problem of copious host-derived sequences, prior investigators have employed host depletion steps. And host depletion is a binary classification task of determining whether a sequence is from a host or not from a host. So instead of this existing pipeline that I had shown before of taking the metagenomic sequencing output and inputting it into a metagenomic classification method that uses a reference database with both host and microbial DNA to perform the classification, we would use the following pipeline, where before inputting the sequences into a metagenomic classification method, we would input those sequences into a host depletion method. that removes the host sequences and then takes those microbial sequences and inputs them into a metagenomic classification method. And in this pipeline, the metagenomic classification method would only need microbial DNA in the reference database because all of those host sequences have been removed. So the benefits of using host depletion for downstream methods are it allows them to be more accurate, faster, and cheaper. More accurate because it gives downstream classification methods a slightly easier classification task. It removes one class that they would need to do the classification on. It also allows these methods to be faster because downstream methods need to classify less sequences. And it allows them to be cheaper because downstream methods don't need to store host genomes. Okay, so previous work in computational host depletion can be broken up into two different categories. The first category are pairwise alignment-based methods, and an example of this is Minimap2. The way that these types of methods work are that they get a query sequence and they align it to the reference genome of interest. And in this case, we want to see if the query sequence is from a human, so we align that query sequence to the human reference genome. In this case, we see there's perfect alignment, so we would output that the sequence is from a human or the human classification label. And if there wasn't perfect alignment, we would output that it wasn't from a human. The other class of methods in computational host depletion are lookup table-based methods, and examples of this are Kraken-2 and centrifuge. And the way that these methods work are they take that same query sequence, but instead of directly aligning it to the human reference genome, they break the query sequence up into k-mers or subsequences of length k. And in this case, we are breaking up the sequence into subsequences of length 3 or trimers, TCG, CGC, and GCC. And then we look for direct matches between those trimers and the trimers that exist in the human reference genome. So in this lookup table, we have all of the trimers that exist in the human reference genome. In this case, we see that these trimers perfectly match these trimers. And so we can output that this query sequence has the human classification label. So the issues with these existing computational host depletion methods are that they can be inaccurate on sequences from error-prone technologies. And this is because they build their reference databases using reference genomes, which are very curated and have very few errors. So when we're doing this direct matching, we're unable to account for a lot of the errors that could appear in error-prone sequences. And these methods are also costly because they require storage and memory to store the reference databases. So why do we think that machine learning would help? So we hypothesized that a machine learning-based approach would be more accurate and cheaper than existing approaches. More accurate because we hypothesized that an ML-based approach would be more robust to sequences from error-prone technologies because we're not directly aligning sequences or k-mers to a reference genome. We can do something a little bit more complicated. And we hypothesized that an ML-based approach would be cheaper than existing approaches because they can use less storage and RAM than existing host depletion methods. And this is because an ML-based approach can learn patterns from raw DNA sequences without the reliance on an external database. But why is DNA classification challenging for machine learning? So ML hasn't been applied to host depletion in past work, but it has been applied to multi-class metagenomic classification and novel pathogen detection. And current approaches in those areas have two issues. First, they can be inaccurate and slow at performing variable length sequence classification, and sequences from error-prone technologies happen to be variable length. They can range from length 100 to over 1,000. And the second issue is that they typically don't build diverse training sets, and they can be inaccurate at classifying sequences that differ from those in its training set. So our contributions are that we developed a novel machine learning-based host depletion method, AMAISE, that can perform variable length sequence classification accurately and quickly on a diverse set of input data. So we first leverage global average pooling to handle the variable length sequences. And we use new training data sources to improve the ability of our model to classify a diverse set of input data. We also rigorously evaluated our approach by providing a comprehensive comparison of our approach to existing alignment and classification methods in their ability to perform host depletion. And we also evaluated our approach as a pre-processing tool applied before downstream microbial taxonomic classification. Okay, so now I'm going to be getting into details of our method. First, starting with AMAISE's architecture, we start with our input to AMAISE, a DNA read. That DNA read first passes through 4 convolutional layers, and these convolutional layers learn a rich feature representation of the DNA read. The next step is passing that feature representation through a global average pooling layer and a fully connected layer to get the probability that a DNA read is from a host. And these global average pooling and fully connected layer aggregate the learned features into that probability. And then the last step is thresholding that probability for the final classification decision. If the probability is below the threshold, we state that the DNA read is from a microbe. If the probability is above a threshold, we state that the DNA read is from a host. Okay, so going back to the issue with existing ML-based approaches for DNA classification, the first issue is existing methods are inaccurate or slow at performing variable length sequence classification. Existing methods either truncate the input or pad the input. And truncation can cause inaccuracy because they remove important parts of the sequence, while padding causes methods to be slow because it causes an increase in classification time since we're adding additional aspects of the input to be classified. To solve these issues with existing methods, we leverage global average pooling to handle the variable length sequence. And this average pooling averages the learned features over the sequence dimension, allowing our method to be agnostic to the length of the sequence. So if you remember from our architecture diagram, the global average pooling occurs after we learn a feature representation of our DNA read via the convolutional layers, and it allows our method to be agnostic to sequence length. So the second issue with existing ML-based approaches are that they don't build diverse training sets, and they can be inaccurate at classifying sequences that differ from those in its training set. Existing methods, like existing computational host depletion methods, train models on sequences from reference genomes. And the issue with this is that these methods are not able to accurately classify sequences from error-prone sequencing technologies. So to solve this, we use new training data sources to improve our learned representations. And instead of just training our model on sequences sampled from reference genomes, we also sampled sequences from sequencing technologies, allowing our method to be more robust to sequences from error-prone technologies. Okay, now I'm going to be getting into the evaluation setup of our method. We had 3 different classes of evaluation metrics that we were looking at, accuracy, speed, and cost efficiency. As I had said before, accuracy is important to ensure that the correct treatment can be assigned. And for our ML method and host depletion comparison, we measured accuracy using classification accuracy, sensitivity, and specificity. Sensitivity is the percentage of sensitivity is the percentage of hosts that were correctly identified, and specificity is the percentage of microbes that were correctly identified. And then for our preprocessing or metagenomic classification comparison, we looked at overall accuracy on the host data and overall accuracy on the microbial data. Then to measure speed, which is important to ensure that the treatment can be given in a timely manner, we measure that using wall clock time. And then for cost and efficiency, which is important to ensure the accessibility of the method and ability of it to be used at the bedside, we measure this using storage or RAM and DRAM usage. And the test sets that we used for our evaluation contain nanopore reads. And this is because nanopore sequencing currently produces the longest reads and is the only platform that produces real-time sequencing data, both of which are useful for clinical diagnostics. And all of our test sets contain human in the host fraction and bacteria and fungi in the microbial fraction. Okay, now I'm going to be getting into our ML model comparison results, which presents a comparison of AMAISE to other machine learning-based solutions that use other architectures and training schemes. And our hypotheses here are that existing methods that concatenate or pad outputs would be improved by using global average pooling, and existing methods that use a single training data source, like just sampling sequences from reference genomes, would be improved by using multiple training data sources. Okay, and for this comparison, we looked at one test set, which contained a human host percentage of 50%. We found that machine learning methods did not, their performance did not change based on the percentage of host DNA in the test set, but non-ML host depletion methods performance did change. So when I show you results from that comparison, we looked at a variety of test sets with different percentages of host in the sample. Okay, so for the first issue of existing methods being inaccurate or slow at performing variable length sequence classification, where we see sensitivity and specificity, we want to be high, and classification time, we want to be low. We see that when we pad the inputs, we have a high accuracy, sensitivity, and specificity, but a high classification time where we want a low classification time. When we concatenate the input, that we have a low classification time, but that comes at the expense of a lower specificity, sensitivity, and accuracy. And in this case, we have peak memory and storage usage held constant, so I don't include that in the table. When we global average pool, we can maintain the high accuracy, sensitivity, and specificity of padding the inputs, but we have a lower classification time. So AMAISE was faster than a model that pads inputs to the maximum length with no decrease in sensitivity and specificity. And AMAISE had a higher sensitivity and specificity than a model that truncates sequences to the minimum length. So for the second issue of existing methods not building a diverse training set and being inaccurate at classifying sequences that differ from those in its training set, we have here models that were trained just on nanopore sequences and just on reference genome sequences sampled just from reference genomes. And we see here that both of these methods have a low accuracy. When we train on just nanopore sequences, we have a high specificity, but a low sensitivity. This flips when we train on reference genomes, we have a high sensitivity, but a low specificity. And we want all of these metrics to be high. When we train on both nanopore and reference genome sequences, we have a high accuracy, sensitivity, and specificity, so are able to achieve the best of both worlds. So training on only reference genomes or only data from a sequencing technology results in lower sensitivity and specificity compared to training on both reference genomes and nanopore data. Okay. So takeaways from this section, we developed a novel machine learning-based host depletion method, AMAISE, that can perform variable length sequence classification accurately and quickly on a diverse set of input data. We leverage global average pooling to handle variable length sequences. And we use new training data sources to improve the ability of our model to classify a diverse set of input data. Okay. Now I'm going to be getting into a comparison of AMAISE to other non-ML based host depletion methods, as well as a comparison of AMAISE plus metagenomic classification methods compared to those methods alone. So first, for our host depletion comparison, we're comparing AMAISE's ability to perform host depletion to other computational host depletion methods. And this is a binary task of determining whether a sequence is from a host or not from a host. Then we have our metagenomic classification comparison, where we're comparing the metagenomic classification pipelines with and without AMAISE, and this is a multi-class task. So our hypotheses here are that first, AMAISE will be more accurate and cheaper than k-mer and alignment-based approaches at performing host depletion. And our second hypothesis is that AMAISE will improve the accuracy and efficiency of metagenomic classification methods. We have 5 test sets that we're looking at for this evaluation, and each test set varied in the percentage of reads of the sample that pertained to host. In our first test set, we have 1% host data, 99% microbial data. The second one has 25% host data, 75% microbial data. The fifth one resembles a gut microbiome with 50% host data, 50% microbial data. The fourth one, we have 75% host, 25% microbial data. And our last one, the most copious amount of host sequences, resembles the lung microbiome with 99% host data and 1% microbial data. And so we used 2 different environments for our evaluation. We evaluated AMAISE on a laptop-like environment, since this applies broadly in resource-constrained settings, but some of our existing approaches, Minimap2 and Kraken2, were not able to run successfully on a laptop environment due to memory and storage constraints. So we also evaluated all methods on a server for a fair comparison. And the only evaluation metric that changed based on computational environment was the speed. So when I present the results, first I'm going to be delving into our server results with all the methods, and then showing you how the speed changed when we moved to laptop-like environment. So first, for our host depletion comparison, where we were comparing AMAISE's ability to perform host depletion to other computational host depletion methods, our hypothesis here is that AMAISE will be more accurate and cheaper than k-mer and alignment-based approaches at performing host depletion. And just as a reminder of the host depletion schematic workflow, we have our of the metagenomic sequencing output of a sample or all of the DNA in that sample. The host depletion method removes the host sequences and outputs the microbial sequences to be used for downstream methods. And the three methods that we're comparing to AMAISE are Kraken-2, centrifuge, and Minimap2. Or if you remember, Kraken-2 and centrifuge are the lookup table-based methods, and Minimap2 is the alignment-based method. Okay, so here are the results of our host depletion comparison. First, I'm going to be talking about our accuracy, sensitivity, and specificity results, where we want all of these metrics to be high. On the y-axis of all of these graphs, we have accuracy, sensitivity, and specificity, and on the x-axis, we have the host fraction varied from 1% to 99%. And AMAISE's performance is in purple, and our baselines are in red, blue, and green. So we see here across all host fractions, AMAISE consistently achieves a higher accuracy here and sensitivity, as well as a comparable specificity compared to the baselines. And it's especially notable that as we increase the host fraction, all of our baseline methods start to decrease in accuracy, while AMAISE's performance remains pretty consistent. Next, looking at storage, where we want all of these metrics to be low, AMAISE is still in purple compared to our baselines in the other colors. AMAISE requires less storage compared to all of our methods and remains competitive with respect to peak memory usage compared to our baselines. So AMAISE is the lowest storage requirements here and has lower peak memory requirements than Minimap2, which has significantly high peak memory usage and remains competitive with respect to Kraken-2 and centrifuge. For our final evaluation metric of classification time, where we also want this to be low, we see that AMAISE in purple, it remains competitive with respect to speed compared to our baselines and is especially faster than centrifuge. We see here that centrifuge starts to get more and more slow as the host fraction, the percentage of host DNA in the sample increases, while AMAISE's performance remains consistent. And we see the same thing happening for Minimap2. Okay, now for the change in performance on a laptop-like environment. As I had said before, the only evaluation metric that changed was speed. Here on the x-axis, we have our methods, and on the y-axis, classification time. And each of these different graphs represents the performance on a different percentage of host compared to microbial data in the sample. We see here that across the board, all methods were slower on the laptop-like environment than on the server. We have in red, these bars are the speed on the laptop-like environment. In blue, we have the speed on the server, and we see that classification time increases. The red bars are always bigger than the blue bars, meaning that our classification time increased on the laptop compared to the server. However, AMAISE still remained faster than centrifuge on all test sets with a host fraction greater than 1%. So when we have 1% host data, AMAISE is the slowest method on the laptop, but as we increase the percentage of host data in our sample, AMAISE starts to get slower or faster than centrifuge, whose classification time continues to increase as we increase the amount of host data in our sample. Okay, now I'm going to be getting into our results of our metagenomic classification comparison, which is a comparison of our metagenomic classification pipelines with and without AMAISE. And our hypothesis here is that AMAISE improves the accuracy and efficiency of metagenomic classification methods. Okay, just as a reminder of the metagenomic classification schematic workflow, we're comparing two different pipelines here. The first is highlighted in orange, where we have our metagenomic sequencing output that we feed into a metagenomic classification method that needs both host and microbial DNA in the reference database to do classification. We're comparing that to this pipeline here, where we input the metagenomic sequencing output into a host depletion method that removes the host sequences and then inputs the microbial sequences into a metagenomic classification method that only needs microbial DNA in the reference database to do the classification. And the methods that we're comparing here are centrifuge with host and microbial DNA in the reference database as our baseline compared to AMAISE plus centrifuge. So AMAISE plus centrifuge with just microbial DNA in the reference database. And we focus our comparison on centrifuge with and without AMAISE, since centrifuge outperformed Kraken-2 on every metric except for classification time. But I have the results for Kraken-2 in an appendix if anyone is interested. Okay, so first I'm going to be showing you the results of host accuracy, where we want higher to be better. And we have AMAISE plus centrifuge in purple, centrifuge in red. We see here that the pipeline that included AMAISE consistently achieved a higher host accuracy compared to the pipeline without AMAISE. And this is across host fraction again on the x-axis, host accuracy here on the y-axis. When we look at cost, where we want that to be lower, peak memory usage specifically, AMAISE reduces the peak memory usage requirements for centrifuge. The pipeline with AMAISE and centrifuge had a lower peak memory usage than centrifuge alone, which is what we want. And then finally, speed, where we also want classification time to be lower. We see that the pipeline with AMAISE's classification time decreases as the percentage of host data in the test set increases, while the pipeline without AMAISE's classification time increased. So when we have 1% host DNA in the sample, AMAISE plus centrifuge is slower than centrifuge, but as we start to increase the percentage of host data in the sample, AMAISE plus centrifuge becomes faster than centrifuge alone. Classification time continues to increase. Okay. So looking at our comparisons on a laptop-like environment, where again, the only evaluation metric that changed was speed, we have here this classification time of our methods on a laptop-like environment in red, and then the classification times of our methods on a server in blue. Again, like the host depletion comparison results that I had shown before, the red bars are always bigger than the blue bars. So all methods ended up being slower on the laptop-like environment than on the server. But AMAISE plus centrifuge remained faster than centrifuge on all test sets with a host fraction above 25%. So if we look at these two data sets, we see that AMAISE plus centrifuge ends up being slower than centrifuge, but as we increase the amount of host data in our sample, AMAISE plus centrifuge starts to become faster than centrifuge, ending here with 99% host data, where AMAISE plus centrifuge is much faster than centrifuge. Okay. So our takeaways from this section: we developed a novel machine learning-based host depletion method, AMAISE, that can perform variable length sequence classification accurately and quickly on a diverse set of input data. And we rigorously evaluated our approach and found that it had higher accuracy and lower storage requirements than other computational depletion methods, and it improved the accuracy and cost effectiveness of downstream metagenomic classification methods. Finally, I'm going to end with some results related to interpreting our machine learning model. So specifically, the question that I want to answer in this section are what k-mers are deemed most important by AMAISE for classification. And a k-mer, as I said in the background section, are substrings of length k. For example, with this sequence, AGC, TCC, the formers are the first four nucleotides in the sequence, AGCT, then GCTC, and then finally CTCC. And the interpretability method that we're using to examine the k-mers that are deemed most important by AMAISE for classification is called deep lift. And this method identifies the importance of input features on a machine learning model's classification. And specifically, on a randomly selected subset of sequences in our test set, we were determining the 15 mers that contributed to AMAISE's classification decision. And we looked at 15 mers because this is the length of subsequence that Kraken-2 uses to perform its classification. So first, I examined the 15 mers that most contributed to a microbial classification, or the way to interpret this histogram is they least contributed to a host classification. So we see the contribution to a host classification label that's negative, which means these sequences contributed to a microbial classification label. So the way to interpret this graph is the histogram shows you how much this entire 15 mer contributed to a microbial classification label. And then these are the top 5 15 mers that contributed most to a microbial classification label. And the nucleotides here are the color around them is dark based on how much they individually contributed to this final score. So the main takeaway that we found from these 5 15-mers in terms of how much they contributed to the microbial classification label is that 4 out of the 5 of them contain a CG dinucleotide. And this is in comparison to when we look at the 15-mers that most contributed to a host classification label, none of them contained that CG dinucleotide. So they all had an absence of the CG dinucleotide, and this is a phenomenon known as CG suppression. And this is a phenomenon that CG dinucleotides are uncommon in vertebrate genomes. So it is encouraging that AMAISE captures known biological phenomena, and this indicates that AMAISE can generalize to classify data from species not present in the training set. We tried to include a variety of species in our training set, but the fact that AMAISE was able to pick up on this general biological phenomena is an indication that AMAISE can generalize to unseen data from what it was trained on. However, this might lead some of you to think that AMAISE is solely relying on CG to classify our inputs, which we found that it's not. Compared to a classifier that only uses the % of CG in a sequence to make its classification decision, AMAISE had a much higher accuracy than that method. We also found that existing k-mer and alignment-based methods rely on CG content to a much larger extent than AMAISE. So these graphs here, we show accuracy, sensitivity, and specificity compared to, on the x-axis, the % of CG in our sequences. And looking at sensitivity and specificity is a little bit more interpretable than the accuracy plots. If we look at our sensitivity plot here, we see that as the % of CG in sequences increases, Kraken Centrifuge and Minimap2's performance significantly drops, while AMAISE's performance remains stable. And this makes sense because there's supposed to be CG suppression in vertebrates. So as there's more % CG in sequences, existing k-mer and alignment-based methods struggle to classify those sequences, while AMAISE is able to classify them because it doesn't rely on the % of CG as much as these methods. And then when we look at specificity, we see that Kraken 2 and Centrifuge struggle to classify sequences with a small amount of CG because there isn't supposed to be CG suppression in microbes, and specificity measures the % of microbes that were correctly identified, but AMAISE is better able to classify those sequences. So k-mer and alignment-based methods rely on CG content to a much larger extent than AMAISE. So looking ahead, accuracy, speed, and memory efficiency are all important in developing bedside clinical diagnostic technologies. High accuracy is important to ensure that appropriate treatments are selected. Speed is important to ensure the treatment can be given in a timely manner. And memory efficiency is important to ensure the technology is broadly accessible. Given the improvements provided by adding AMAISE to the current metagenomic classification pipeline, we believe that this work brings us one step closer to clinical diagnostics via DNA sequencing technologies at the bedside. So here are some final takeaways, a link to my paper, my contact information, if you would like to ask me any questions via email, as well as the funding acknowledgements for this project. Thank you.
So if anyone online has questions, okay, if anyone online has questions, feel free to put them in the chat box or raise your Zoom hand. And if anyone in the room has questions, feel free to ask.
I'm gonna start with you.
Great talk. Thank you very much. So just to clarify, you guys used nanopore sequence data to for the model, right?
Mhm.
So how would you be able to apply other sequencing technologies like short read, like to the model as well?
Yeah, so since AMAISE was trained on base called reads, it applies broadly to any input sequence. I think to ensure that it would be, I guess, the most accurate, I would recommend retraining AMAISE to include, I guess, whatever specific sequencing technology that you would want to apply it to. But the fact that AMAISE was able to learn, I guess, the presence of CG is indication that it could potentially apply broadly to other sequences from other sequencing technologies, but to ensure highest accuracy, I would recommend retraining or adding those sequences to the training set.
Yeah, I have a question about where you may have gotten your ground truth labels to train AMAISE. So, it's I'll just make an analogy to explain this. I'm like doing a single cell. For example, a common task might be to do classification, but it's often wrong to train your ML model on labels that was already labeled by like some earlier ML method that labeled the cell type. So
Right.
you're kind of training on something that was already machine generated and
Uh-huh.
human annotated. So you need to kind of, it's better if a human can actually say like, oh, this cell was actually cell type ABC or whatever. So here, like the labels must have come from somewhere for you to train a maze. I was wondering what that process was.
Yes, yeah. So the test sets that I presented here were synthetic microbiomes that we created. So we got the I think there's a human reference standard that was created in 2018 by some research group where I guess they had confirmed that all of the sequences in the data that they provided were from a human. So we used that. And then we got our bacteria and fungal DNA in the same way. So these were artificially created microbiomes based on sequences that were already confirmed to have that label. In our paper, we have results on a real microbiome where we got the labels, I think, using Minimap. But for all of the results that I showed here, they were artificially created microbiomes based on sequences where we knew what the true label was.
And I have a question. Second question was about so AMAISE was working really well on different percentages of the host DNA. I was wondering in your training set, you had like some more like details on imbalance training or are they usually like for example, yeah, I mean, yeah, if you have some like a real sample where you expect there to be like 99% human, and so like how did you deal with that kind of imbalance to improve AMAISE's performance like this?
Right. So our training set had 50% host, 50% microbial DNA in it. But I think why AMAISE was able to succeed so much more on the test sets with a large human fraction is that we included the reference standard that we trained on for AMAISE from a nanopore sequencing technology, I think allowed to generalize much more than existing methods. I think that our crack-induced centrifuge Minimap struggled much more on the noisy host sequences specifically. And I think it's because a lot of them contained a lot of CG. As you can see, like existing methods tank significantly more in terms of sensitivity when the percent of CG increases. And this measures our accuracy on the host sequences. So I think for some reason, I think because the host sequences looked so different from what existing methods were expecting, and AMAISE was able to deal with that because we were able to train on nanopore sequences directly. That's what led to a success more than class imbalance.
So there are a couple of questions that have come in online. The first is from Travis, who said, it seems that current methods of truncating or padding to equalize input dimensions before inputting into CNNs. In your architecture diagram, it seemed the global average pooling happened after the CNN. Am I understanding that correctly? Can your CNNs use inputs with non-equal dimensions?
Yes, yeah. I think, yeah. So I guess the way that we handled that was that the way that you could handle that was input each sequence individually. So that because I think if you input sequences in a batch, they have to be the same length in order for, I guess, you to input it as a matrix, but inputting sequences one by one, AMAISE can handle any sequence of any length.
Great. And then the second question that came on, and then we can go back to questions in the room. This is from Patricia, who said, excellent presentation. Good job making this clear and logical. Could you say a little more about the choice of deep lift and the alternatives that were considered?
Yeah, so the reason that we selected deep lift is because it was designed with DNA sequences in mind. Specifically, I think a lot of other interpretability-based methods are for images or for other inputs. The fact that deep lift was designed to interpret DNA sequences already, and some existing work had already been done in selecting. So the way that deep lift works is that it compares the output of your method to the output related to a reference input. And there were a lot of experiments done on the appropriate reference to use for DNA-based methods. So that's why we chose to use deep lift. I think there are a ton of different interpretability-based methods that could be used to analyze AMAISE, but because deep lift had already been looked at in the context of DNA, that's why we chose it.
Thank you.
For the microbial, group together bacteria and fungi, but fungi are eukaryotic. And I would picture that they might be more similar to those bacteria. I was curious if you had the bias or the predictive accuracy separated out between the bacteria, microbes, and the eukaryotic bacteria.
That's a great question. I don't have those results, but I think I remember qualitatively looking at our output and we struggled more on fungi. So I think that was kind of across the board for all methods. I don't know particularly which method struggled more or less. That would be something that I should look into. Results.
You said, for the reason analysis or analysis to stand out is basically we look at this global average. So can you elaborate more on that?
Yeah, so it's not, I think, something that's typically done, this sort of, I guess, pooling over the entire sequencing dimension. And I think it's because in most cases, machine learning inputs are fixed in size. And so I think that just like I guess there's this thought that, well, why would you need to be agnostic to size because all of your inputs are going to be the same size? I don't think that this is a problem that comes up much in machine learning. But I think we found that, I guess, being agnostic to sequencing size allowed us to, I guess the biggest issue with truncation is that you're losing out on valuable parts of the sequence that could be important for accuracy. And the other fix for that was to use padding. But the issue is that nanopore sequences particularly can range in length so significantly that if you pad to the maximum length, you're increasing your classification time a lot. So we found that doing this global average pooling allowed us to have kind of the best of both worlds of using all of the input, but not being significantly slower on smaller inputs.
So I forgot which slide it was, but you had one where you were looking at the time performance of the different proportions of like hosts and microbials sequences. I was wondering. Oh yeah, actually no.
Is it this one or?
No, the one before.
Okay.
Actually, yeah, I was just gonna ask, like, if you could talk a bit more about sort of the variation between like model performance of like different fractions and like whether, because I know the sort of big the main example you gave at the beginning was like a sample that is like mostly hosts, and only part or only a small bit microbial. Just wondering about the inverse of that, because I know that's a thing for a lot of like microbial like servers.
Yeah, so I think so. I guess the reason that non-ML based approaches, and specifically, I guess, centrifuge and minimap here struggle when there's a lot of host DNA in the sample compared to microbial, is that host reference genomes are a lot bigger than microbial reference genomes. So pulling that into memory and looking over the host reference genomes to perform the classification takes longer. So if there are more host sequences, you have to do that more often. And so those methods can struggle versus the inverse of when there's a lot of microbial sequences relative to the host, the reference genomes you have to pull into memory are much smaller. So that takes less time. So I think that existing methods are optimized on I guess, a speed basis for kind of the index of a lot of host sequence or a lot of microbial sequences relative to the host sequences. But as the host fraction increases, those methods start to break down. And so we see that AMAISE is probably the most useful in cases where you're analyzing a microbiome like the lung microbiome with a significant number of host sequences, when you have the inverse of a lot of microbial sequences relative to the host. If all you care about is speed, I think that existing non-ML based approaches are also good, except for even though centrifuge is a little bit slower than AMAISE.
Thank you.
I thought your GC analysis was really interesting. It got me thinking that perhaps there is this easily identified patterns that separates them, and so the model could be very compact. Like it doesn't need to look at very much to tell. But I guess sort of on the other end, you can picture, gosh, they're virtually indistinguishable. So just has to memorize all the sequences and you're back to the reference genome point. I was curious if you had thought about sort of assessing where you're up in that spectrum of memorizing versus like having really crisp lines, and maybe in terms of like model capacity, like how does your performance degrade as you get smaller and smaller models? And then also maybe thinking about trying to not just distinguish humans from microbes, but like microbes from microbes or other sort of more challenging types of discrimination paths.
Totally, yeah. So we actually started this project as trying to replace the entire pipeline of multi-class metagenomic classification. And we found that existing methods are so successful because I think when you get to these fine-grained differences, you have to do exact matching, and it's not as easy to extrapolate patterns as it is with this kind of simpler host versus not host test. So you totally hit the nail on the head. And I think we found that machine learning right now, in terms of speed, accuracy, and efficiency perspective, works best as sort of this kind of higher level separation, and existing methods, I think, are still superior when we do more fine-grained differences.
Yeah. Any other questions?
Yeah, so when you build up your training data set, how did you construct your host plans? Meaning, like, I know that it's not from a real person, but like, do you have any background on what type of people are those like genomes are from? Is it covered enough like diversity to represent the entire population that you don't introduce any bias towards this host type you're using?
Right. So we included 3 host genomes in our host fraction, or sampled sequences from 3 different hosts. We did human, pig, and mouse. And we included some human nanopore sequences, but we didn't include any pig or mouse nanopore sequences. And I have results in the full paper if you're interested on performance with nanopore, pig, and mouse sequences. And we found that we could do well on those sequences. So we were able to extrapolate from the human nanopore data, patterns that exist in nanopore data, but we're able to do well on pig and mouse because we included that in the reference genome aspect of our set.
Yeah, so nanopore has a very neat feature, which is like technology allows it to read in a sequence and determine if it's what it's looking for, and if not, back it out.
Yes,
and
yeah.
back again. So it's possible to apply or like real time and labeling in the sequence while it's getting a read and deciding whether to continue or no.
So I guess the issue is that we like the additional step we would need to do is be base calling in real time, because I think a lot of existing nanopore specific approaches work on base called electric signal. We work on base call signal. So if you could base call in real time, we can work in real time. Yeah, I guess, but the issue with methods that work on the electrical signal is that they're not accessible to other sequencing technologies. We see sort of AMAISE, kind of the framework of AMAISE could work
Right.
on PacBio, on
Without changing
Illumina.
their entire model. though.
Yes, yeah.
Kind of goes back to my first question about like using sequence technology, but I was wondering if it would be possible to train on like like instead of just like focusing on like one sequence technology or like one like having like a nanopore specific model or Illumina specific model, if there would be any complications with just using a variety of sequence technologies to train just like the model together.
Yeah, I guess I don't know what the trade-off would be in terms of accuracy on each individual sequencing technology compared to overall. We tried to do that with, I guess we started this project thinking of applying AMAISE to both short and long reads. I think the issue with short reads is because they have very low error and they're very short. Existing approaches, Kraken centrifuge minimap, are designed to operate well on short reads that have very low errors, so we weren't able to do better than them. I think that a machine learning based approach works is kind of the best in comparison to existing approaches on long reads. So I think actually I mean we haven't done any experiments on this, but I think AMAISE would probably work well kind of out of the box on just PacBio sequences. But I think when you look to apply it on short reads, I think machine learning might not be most optimal to handle that. Yeah.